Hello students, today let's study regarding DNA replication errors. DNA replication is highly accurate process but still errors can occur. These errors are caused because of the various reasons like misincorporation of nucleotides, second is base pairing, mismatches, third is failure of proofreading mechanisms, fourth is DNA slippage. DNA slippage is a phenomenon which causes insertions or deletions in DNA sequence during replication. We'll read this in detail. Okay. Now let's look into the DNA replication errors. The first one is base pairing mismatches. During replications, there can be instances where wrong nucleotides pairs with its counterparts. Wrong nucleotide pairs with its counterpart. That is, for example, adenine has to pair with thymine, but instead of pairing with thymine, adenine goes and pairs with guanine. Okay, what is the consequences of this? Mismatch base pairs can lead to mutations if it's not corrected. Okay, now let's look into the second type. The second type is misincorporation of nucleotides. Here the DNA polymerase which is responsible for synthesizing new DNA strands might occasionally insert. This can insert an incorrect nucleotide. We know that DNA polymerase has proofreading mechanism. DNA polymerase has a built-in proofreading function that allows them to backtrack when it is synthesizing new strand of DNA in 5' to 3' direction. It can go and check back the wrong nucleotides if it has attached any in the new strand, and this phenomena is called as proofreading mechanism. So it backtracks and fixes these errors by removing this wrong nucleotide and replacing it with the correct one. Okay. Which enzyme does it? It is DNA polymerase enzyme. Now let's look into the another phenomena, that is, now let's look into the phenomena called DNA slippage. DNA slippage is a phenomena which brings changes in the DNA either by insertion or deletion of nucleotide. Okay, there are various phenomena in this. First, let's look at IDLs, that is insertion, deletion loop. Slippage can occur when one DNA strand temporarily moves out of the alignment with another, creating a loop. So, it occurs when one DNA strand temporarily moves out of the alignment with other, creating a loop. Okay, here the slippage is occurring in the new strand. What could be the consequences of this? It can result in addition or deletion of one or more, addition or deletion of one or more nucleotides, leading to the variations in the DNA. Okay. Insertion deletion loop happens when one of the strands moves out of alignment with another, creating a loop, and this results in the formation of a slippage. Okay, which helps in addition or deletion of one or more nucleotides, causing the variation in the DNA. Okay, the second type which results in slippage are microsatellites. Microsatellites are the regions in DNA. They are the regions in DNA with short repeated sequence. For example, it can be CA, CA, CA. What is cysteine? Adenine, cysteine, adenine, cysteine, adenine. Such a short repeated sequence can be called as a microsatellite. Okay. What will happen when microsatellite is present? Due to their repetitive nature, they have a repetitive nature. These regions are prone to slippage during replication. Okay, the consequences are that slippage in microsatellites can cause changes in number of repeats, leading to genetic variation. Okay, now the third type of slippage can be because of DNA polymerase slippage. During replication of repetitive sequence, replication. Okay, when repetitive sequence is undergoing replication, DNA polymerase can sometimes slip or misalign, leading to errors. The slippage in this can result in, this causes a slippage, and this results in addition or deletion of nucleotides, thus error in DNA. Okay. So we have learned the various types of slippage. We have understood what slippage is, what various types of errors that could occur in replication. Okay. Now, what are the various correction mechanisms? Okay. Correction mechanisms of DNA replication. So, what are the various correction mechanisms? So, let's look in each one of it. So, first one is proof reading. The best enzyme for proofreading is DNA polymerase 3. They have the ability to proofread function that allows them to correct errors in real time. Real time correction of errors. So, as soon as replication is starting synthesizing new strands, at that time only it will backtrack and correct any error that is occurring. If mismatch or misincorporation occurs, the DNA polymerase can reverse its direction, move in the opposite direction, remove the incorrect nucleotide and replace it with the correct one. Okay, the proofreading mechanism by the DNA polymerase 3 enzyme. Okay, the second one is mismatch repair. The mismatch repair is done when the DNA replication is complete. Proofreading is done while DNA replication is happening. Mismatch repair is done when DNA replication is complete. Mismatch mechanisms, repair mechanisms scan newly formed DNA strand. Okay. If mismatch is detected, then incorrect nucleotide is removed and replaced by correct nucleotide. So, here we have to understand that mismatch repair mechanism is done after the new strand is completely synthesized, whereas proofreading mechanism by DNA polymerase is carried out during the synthesis of the DNA strand. Okay, it is done in the real time. Which one? The proofreading mechanism. Okay, now let's move to the third one, that is slippage correction. So, what do you mean by slippage correction? Repair mechanisms are in place to correct errors resulting from the slippage that is caused during DNA replication. We have read different types of slippage occurring. Okay, so whenever there is a slippage occurred, DNA repair mechanisms come in its place and corrects this slippage. So, the mechanisms that detect and fix addition, so it can be stated as the mechanism that detects and fix additions or deletions of nucleotides that occur during slippage. Okay. So, these are the three correction mechanisms. Overall, we can state that DNA replication is highly accurate process, yet it has errors. And the cell has various mechanisms to deal with these errors. Understanding these processes at detailed levels helps us to understand how efficiently our system works to transfer the genetic information from one generation of the cells to the another generations without being altered. So thank you friends. Here we done with the DNA replication errors and the slippage mechanism.